Another nutrient that we looked at during the march was phosphate, and what we did with this was we placed 10 milliliters of sample that we collected from the marsh, and then we placed it into each bottle into a continually plastic centrifuge tube. Then we added mixture reagents and then mixed. Then we placed these samples into a spectrophotometer, and then we read their absorptions. For our conclusion, our results, we don't have them yet because it's an ongoing project. And but so far, the Piermont Marsh is dominated by Phagmites australis and yet it forms valuable habitat for necton and vertebrates. The highest density of fish population occurred in interior pools. The type of fish that we collected was one that is Sheryl, Reyes, and 90% of this was caught while we were in the marsh. Marsh fauna showed non-random movement and soon out of the marsh correlated with tidal cycles. Migratory behavior is strongly influenced by hydrology and geomorphology of the marsh.
Since it's an environmental experiment, in the future we want to find out how the fish and the nutrients go back and forth between the streams and the interior ponds. And also it's possible to have a healthy wetlands in an industrial populated area. And we also want to show that this is a model for what we want to do with other marshes along the metropolitan area in the future. These are our representatives. And we want to acknowledge Harlem University, the Vermont Authority Observatory, Robert Newton, Susan Vincent, Maria, Mike, which is here, lab staff, and co-mentors, Dr. Sadat, ACS students, and staff.
Okay, so once we go out into the marsh, we assemble 3 minnow traps and then attach it to a 3-meter rope. And then once we're out there, we take the traps out, whether they're assembled or not, and we're in the canoe and we come out and we walk in the marsh and we attach it to the rope to the PVC pipe right here, which is one of those flooding things. We measure the water depth in centimeters. And then once we're out there, somebody in the canoe takes this YSI, it can't tell, right here. And we measure temperature, salinity, and dissolved oxygen. Once we're in the boat, we, after either 24 hours or 12 hours, depending on what we're doing that day, we take out the fish and we put them in jars and we kill them right away with formalin. Okay? And then, we'll continue with that. And then we just rinse them and put them in 50% isopropyl alcohol. And then we identify, we weigh, and we measure. Then we report our results.
Okay, thank you.
Questions?
I wasn't too clear on your objectives for the hydrographic sampling. Could you please clarify that?
Which one were you unsure about? Both? We wanted to figure out the phosphate, nitrate, and nitrite levels in each of the sites. The ones we had to mark with the PVC pipes. And we wanted to see how the tide affects nutrients in the creek. That's why we had, like Nunny said, the 24 and 12 hour cycle. Okay. Any other questions?
Can you go back to your code?
Alright. Could you revisit how the Phragmites australis affected the marsh?
Raises the substrate above the water level. It raises the elevation level of the goat. So it controls how many times a month it gets flooded, and how often it's flooded, and how long it's flooded, like how many hours duration.
Like prior to the invasion of the Phragmites, the marsh was actually a lot lower. So when the Phragmites began to raise the substrate with the cones, which is the monk that you see, now the interior floods 10 to 12 times a month instead of every day.
don't know about the rest of you guys, but from what I see from these students, they're all really hardworking. They all want to put the best foot forward and do the best job possible. And really, they really helped along with the research. This is an ongoing project that's going to take probably at least 10 years to complete. But with the research that they did, it helps a lot. And the fact that they were able to understand everything that they did and put together this project along, I'm sure the rest of you, I'm sure you guys learned a lot. I'm sure your mentors learned a lot from you. I just want to thank you all for taking part in this program.